Hi young children, in the previous class we discussed about innate or the inborn immunity where four barriers operate in order to provide us this particular immunity. And the barriers were the physical, the physiological, the cellular and the cytokine barrier. Today we will be studying about acquired immunity. Acquired immunity is something that we acquire in our lifetime. And just like the inborn immunity, it is functional based on four concepts. Let us discuss what these concepts are. The first concept is the recognition between self and non-self. Recognition of self and non-self. Now what is the difference between self and non-self? Any substance which enters into our body, which our immune system fails to recognize as our own self, is known as a non-self substance. I hope it's clear. Substances which enter, which is not recognized, are called as non-self substances. These non-self substances are also called as antigen. The word antigen can be broken as anti and gen, literally meaning antibody generating substances. And these antigens or the non-self substances can range from pathogens to some food materials. For example, some of you are allergic to say egg white or to prawns or to peanuts. So that is also an antigen. So pathogens, food material, blood which is transfused from another person, or it could be a drug. A chemical that we take as a medicine is known as a drug. A drug functions as an antigen. It could also be a tissue which is transplanted or an organ which is transplanted. So antigens are a wide variety of non-self substances ranging from food, pathogens, drugs as well as blood, tissues and organ. So when your body fails to recognize self from non-self, that is the first concept of the acquired immune system. Let us see what is the second concept. The second concept is known as diversity. Now what is the meaning of diversity? Please understand that there are possibilities of millions of antigens to enter into our body. From birth, throughout our life, we are exposed to so many non-self substances or antigens. And this is known as the diversity aspect of our immune system. The immune system is capable of recognizing all these millions of antigens we are exposed to. Suppose we are exposed to a common cold, a virus today. Our immune system immediately recognizes it as non-self. So this is the meaning of diversity. Our immune system can definitely recognize the diverse number of antigens which enter into our body. Let us now see what is the third concept. The third concept is known as specificity. Specificity means out of the millions of antigens which enter into our body, our immune system is so smart that it can easily distinguish between two very similar antigens that enter into body. Meaning, for example, the cowpox virus and the smallpox virus are very similar. Maybe there are only very small molecular differences in that particular virus. Though it is very similar, our immune system can definitely distinguish the difference between these two viruses. Now think about a twin, an identical twin. Now, as far as I am concerned, I find it very difficult to differentiate between two twins. I mean, a pair of twins. When they come in, I always get confused. Is it Kavya or is it Kirti? Or is it Achu or is it Ammu? Now, I get confused because they look so identical. But not so with our immune system. Our immune system is capable of distinguishing even very minute changes. It could be a molecular change or a structural change in an antigen which enters into our body. And that particular concept of the immune system is known as specificity. So recognition of self from non-self, diversity, specificity. The fourth concept of our acquired immune system is memory. Now, what do you mean by memory? Memory is the unique ability of the immune system to recognize an antigen after a long period of time and to produce antibodies against that particular antigen. Now, you may recollect that when you all were very young, or you may have heard that you have taken a polio, oral polio vaccine. Now, this oral polio vaccine is actually an antigen. But even after a long period of time, even now, your immune system recognizes that particular virus. Suppose you are exposed to a virulent form of that virus, your body can immediately produce antibodies against polio. So this is known as memory. So memory is the unique ability to produce antibodies, to recognize an antigen after a long period of time and to produce antibodies. Now let's see how it functions. When your body is exposed to an antigen for the first time, okay, the first time you are exposed to an antigen, your immune system produces a very mild, low intensity immunoresponse. It's very mild and low intensity and it has a short duration. But immediately the pathogen, suppose it's a pathogen which is entering as an antigen, the pathogen is destroyed. This mild low intensity immunoresponse is known as the primary immunoresponse. So the first exposure produces a primary immunoresponse. Now let us see what happens at a later stage. Suppose after a long period of time, your body is exposed to the same antigen. Remember, your immune system has the memory. Immediately it will think, oh yes, I know what this antigen is. And then your immune system produces a very high intensity, long lasting, okay, it's a long lasting high intensity immunoresponse which is called as the secondary immunoresponse. Secondary or anamnestic. It is known as secondary or anamnestic immunoresponse. So this kind of immunoresponse is because your body has that unique power called as the memory. Your immune system has the unique ability called as memory. So these are the four concepts based on which our immune system functions. First is recognition of self from non-self. Second is the diversity. Third is specificity. And the fourth is memory. Let us see the development of the immune system. The immune system develops at a fetal stage. But after birth, there are some stem cells. And these stem cells in the bone marrow, the stem cells are found in the bone marrow. These stem cells undergo a very complex process of differentiation. Stem cells are actually undifferentiated cells. These stem cells undergo a complex process of differentiation and they are converted into a particular form of WBCs called as lymphocytes. I am sure you have learned about lymphocytes. Now these lymphocytes now follow two pathways. Some of them migrate to the thymus and the others remain in the bone marrow. Now let us see what happens next. Those which migrate to the thymus, now thymus is a lobular gland which is seen just below the sternum. This is a sternum or the breastbone. Just below the sternum, close to the heart, it's a triangular shaped gland known as the thymus. This thymus is large at birth but gradually degenerates or atrophies as we grow older. Now the the lymphocytes which migrate to the thymus, within the thymus they will start modifying and they are converted into the T cells or the T lymphocytes. The T lymphocytes are responsible for imparting the cell-mediated immune system or the CMIS. So the T cells or the T lymphocytes impart an immunoresponse in a body called as a cell-mediated immune system. It actually forms a system, cell-mediated immune system. Now what happens to the rest of the lymphocytes? The rest of the lymphocytes remain in the bone marrow, and within the bone marrow they modify themselves as B cells or the B lymphocytes. Now these B lymphocytes impart what is known as the antibody mediated immune system. Antibody mediated immune system, or it is also called as the HMIS, the humoral mediated immune system. So T cells are responsible for the cell mediated immune system and the B cells are responsible for the antibody mediated immune system.